Assalamu alaikum guys again. So today what we are going to discuss is our next topic in the course content. Hopefully the screen is in front of you guys. So the topic that we are going to discuss today is introduction to molecular biology or introduction to molecular genetics. As I told you guys that in this 412 we'll be discussing about some information regarding the nucleic acid molecule, which are DNA and RNA, and some of the basic processes involved with the multiplication of the DNA and how the genetic information is replicated and so on and so forth. So before we jump on to those topic of replication and other molecular biology processes, I thought it's important to give you a basic overview, a general overview of what actually molecular biology is, so that you will have a basic understanding. And because of that basic understanding, you will be more comfortable in understanding the processes of molecular biology in a better way. So let's start our discussion. How we define molecular biology. Now, molecular biology is a branch that deals with the study of biology or biological processes at a molecular level. Now, what is actually a molecular level? You might have heard about the different levels at which the studies are being performed. For example, when you are going to study at the level of an organism, you call it organismal level. Then you can go at the tissue level, then you can go at the cellular level, and then we have the subcellular level as well. So if you go on in this direction, there are molecules within the cell like DNA, like proteins, etc. So the study at that level of the molecule is referred as molecular biology. So specifically, it is a branch which deals with the study of gene structure and the function of the gene at the molecular level to understand how the genetic variation takes place, how the genetic information is transmitted from one generation to another generation, that is called the hereditary, and what is the expression pattern of the genes. So in generally, in this molecular biology, we'll be going through the gene structure, how the genes are expressed, how the genetic variation takes place, how the DNA replication takes place, and so on and so forth. So the molecular biology field overlaps with the other disciplines and other areas, particularly genetics and biochemistry. So going further in detail, let's start with the first thing to discuss. What is genome? Now, the word genome is a term used for an organism in its totality of the genetic information, which is either encoded primarily in the form of the DNA, or in some cases, it could be RNA as well. Like you might be aware of the fact that there are certain viruses which are RNA viruses. The most common nowadays is the COVID-19, which you all are aware of. So the term genome is the total genetic information of an organism. So if it's a single cell organism, it will have, all the genes within that cell. Or if it's a multicellular organism like us, it would be the totality of all the genetic information which is encoded on the DNA, or in cases as I in RNA, in total of the organism. So you can see that picture which is explaining that this is particularly area what we call the gene, having specific codes. The gap between the two genes is called the intergenic region. Then the genes are located on the DNA. The DNA are associated with the chromosomes, and then the chromosomes in totality leads to the genome of an organism. Now, there's a term called genome database. Now, what is genome database? Genome database is a science where we collected all the genetic information of different organisms and we kept it for our own reference. Now, in order to make it more accessible, we divide that database in different groups, and these groups are, based on the simplicity to the complexity level. So the genome database that we have had, it might not be a very recent stats. You can check on the Google for the recent stats. Just here, I'm just going to tell you about what genome database is. So the database is organized in six major organisms group. That includes eukaryotes, including human, bacteria, archaea, viruses, and then below or smaller than viruses are the viroids and plasmids. So you can see in the picture that it's a rough distribution of different groups and the size of their genome database. And obviously, with the inclusion of more and more organisms, this genome database is increasing. So as I said, that it's not the recent stats, but just being here to give you an idea that how the genome database is organized. Now, all living things are primarily grouped in three domains. There could be, other classification in other books, but in generally we classify them in three domains as eukaryotes, prokaryotes, and archaea. So we'll be going through them very quickly, a little bit of description about these three groups of the life. Before we jump on to them, we know that the cell is the basic unit. We all know that cell is the basic structural and the functional unit of every living organism. And, the cell itself is an organism when it comes to a unicellular organism, and then we have, complex organisms, the multicellular organisms. So cell is the smallest living unit, the basic structural and functional unit, and in some cases like bacteria or other unicellular, it is only a cell that is referred to the organism itself. So cells are stacked together on the basis of their structure and on the basis of their function, and that give rise to tissues, and the combination of the tissues give rise to organs, and then from organs, if you go further, it's like organ system and so on and so forth. The cells would have different shapes, like if you compare the cell of a nervous system is different in shape from the cell of a kidney. A cell of a kidney is different in shape from cell in the muscles. So depending upon the function and the structure, they might have different shapes. When it comes to the functions of the cell, there could be a long list of the functions, but here few of the functions have been mentioned, like secretion, secretion of hormones, secretion of enzymes. Cells also function as a storage organ, like they store carbs, they store fats. Cells also have a memory and intelligence when it comes to like the brain cells. Then cells may contract and relax when it comes to muscle cells. They may have a protective function like the skin cells, and they may have a defense role in the body like white blood cells. So as I said, that there's a long list of the functions, but these are few of the functions listed in front of you. Okay, let's start with the eukaryotic cell first. And I'm pretty sure that you guys are aware of these things, so that's why I'm very quickly going through that, just part of intro. The eukaryotes are the advanced, cells. They are more advanced than prokaryotes, and the term "eu" refers to, developed. So there are many unicellular organisms which can be eukaryotic, but most of the time, the eukaryotic cells refer to multicellular organisms. So they are the cells which are found in animals, plants, fungi, and in protista cells as well. Now here is a picture in front of you for a eukaryotic cell. It's a typical picture. You have seen that in many books. By definition, a eukaryotic cell is a eukaryotic cell based on the presence of a true nucleus. Now, what do you mean by the true nucleus? The true nucleus means that the cell does have a genetic material, and that genetic material is distinctly separated by a membrane, and that particular structure is called as the nucleus. So, the genetic material is within the cell and it's being surrounded by a particular membrane, and that particular membrane give rise to the formation of nucleus. So in case of eukaryotic genome, definitely it's going to be more complex when you compare that from the prokaryotes, and it's more bigger. Eukaryotic genetic material could be most of the time would be the DNA, and the DNA in the eukaryotes is linear in structure. Now, this eukaryotic DNA does exist in the form of a complex, and they are normally complex with the protein molecules, which we call the histones. And they have a different role, we'll come to that later on, but just here, just to give them stability. Beside that distinctive nucleus, eukaryotic cells also have other membrane-bound organelles, and they do have a complex internal structure as compared to the prokaryotes. Another important thing about the eukaryotic cells is that they divide by the process of mitosis. In contrast to that, the prokaryotic cells, they are unicellular organism-based cells available, in everywhere in all environments. Examples include bacteria, archaea, etc. Typically, the prokaryotic cells that don't have a nucleus, but that doesn't mean that they don't have a genetic material. Having a genetic material is the basic requirement of an organism. So they do have genetic material, but there is no distinctive membrane that separates it from the rest of the cell. So that's why we say that there is no nucleus. They don't have membrane-bound organelles. Their DNA, instead of the eukaryotic DNA, which was linear, it's circular, and it's contained in the cytoplasm. Their DNA is not supported or bound by the histones as in case of eukaryotes, and that's why we call it their DNA is naked. Their structure is simple, and when it comes to the division, they undergo the division by simple binary fission method. The third group of the life or organism, beside eukaryotes and prokaryotes, are archaea. Now, archaea are definitely less developed than prokaryotes. And in some books, you may find out that archaea is being classified as prokaryotes as well. Like you can see the definition. Archaea are the prokaryote organisms without nucleus, but some aspect of their molecular biology are more similar to those of the eukaryotes. So I would say that archaea is a category which lie in between the pro and the eukaryotes. Like they have some characteristics like prokaryotes, which is without nucleus, but they do have some molecular biology similarity with the eukaryotes as well. Now, in a eukaryotic cell, there is a pathway that eukaryotic cells follow, and that particular pathway is called as eukaryotic cell cycle. And this cycle actually refers to when the eukaryotic cells undergo multiplication, or you can say that when they undergo division. So eukaryotic cell cycle has got different stages, as you can see in the picture as well. It's being divided into four stages as G1, S, G2, and M. And I already have mentioned that the eukaryotic cell division is by the process of mitosis. So D1 is actually the growth and the preparatory phase of the cell cycle, where the cell is undergoing growth and undergoing the preparation for the process of replication or the process of mitosis. So G1 is specifically growth and preparation of the chromosomes for mitosis. S is the synthesis of the DNA. G2 is the preparation phase of mitosis, and M is the final stage where the cell will undergo multiplication, or cell will undergo replication. Now, this picture is a very important picture in molecular biology, and we have a title for this as Central Dogma of Molecular Biology. Now, what does central dogma mean? Central dogma of the molecular biology means when we talk about molecular biology, this picture is giving you the central concept of molecular biology, the central idea of molecular biology. Or in other words, this picture is going to summarize the entire processes involved in molecular biology. So that's why we call the flow of genetic information, and that is being referred as central dogma of molecular biology. Now let's go start from this side of left side of the replication. Now what is replication? We know that replication is the multiplication of the DNA. Like we have one copy of the DNA, and as a result of the replication, we can have two, and then this process will go on, and we have got more and more multiple copies of the DNA. That's called replication. And you can see this; it's a reversible process. You can see this arrow, right? Then from the DNA, we can have synthesis of RNA, specifically mRNA, which is called as messenger RNA. This process is being called as transcription, or in short, you can also call it RNA synthesis. Now, this RNA will undergo further process, and that process will take place on the ribosomes, which are in the cytoplasm, and that process is called as translation, or it can be called as protein synthesis. And as a result of that process, what we are getting as a product is actually proteins. So if we start from the replication and we end up at the protein, this entire process is being referred to as central dogma of molecular biology. Okay, let's start to go with the discussion of the molecule of genetic information, which are the central part of molecular biology topic. And we are going to start with the molecule which is the most significant when it comes to genetic information storage, and that's DNA, deoxyribonucleic acid. Now, deoxyribonucleic acid, DNA, is the genetic material for all cellular organisms and most of the viruses. So this is very clear that when it comes to the cellular organisms, their genetic material or storage of their hereditary information is on the DNA. And in many cases, the viruses, they do have DNA as their genetic material. But we know that viruses are being classified as DNA viruses and RNA viruses. So definitely some of the viruses which are being classified as RNA viruses, they do have RNA in place of the DNA. This DNA is a gigantic molecule which is used to encode the genetic information for all life on the earth. Now, why we call it a gigantic molecule? Uh, in terms of its complexity, in terms of the storage and information it does have, and in real term, it is a big molecule. It is something really significant. So this is the diagram of a eukaryotic cell, and I'm not going in the detail. You are already aware of these organelles and all these things, so I'm not going in the detail. Now, there are some interesting facts and figures about DNA, and it refers to why we call it a complex and a gigantic molecule. Let's talk about the human body. On average, in a human body, there are trillions of cells. And if we go in detail, each cell would have 46 chromosomes. We all know that, right? So every chromosome would have one DNA on it. So according to the research, we found that the DNA is attached with the chromosome in a specific arrangement, what we call the supercoiling. It's very compressed and it's very condensed. So accordingly, if we have got 46 chromosomes, so that we would have 46 DNA molecule. I'm talking about one cell. And if these 46 DNA molecule in a cell, they are stretched. Stretch means they are taken out of that supercoiling. And if we extend them and stretch them and altogether add them, the length of the DNA molecule would be around 2 meters. So now you can think about, look at the size of the cell and then look at the length of the DNA. So definitely it requires a very sophisticated process of supercoiling that would result in arrangement of that DNA within the cell, and not only cell but within the nucleus. So that's how it gives you an idea about the complexity level of the DNA molecule. Then comes the chromosome. For the chromosome, they are, specific number of the chromosomes with reference to a species, and that's why the number of chromosomes present in a cell is being referred to as species number. So for the human cell, the species number, we know it's 46, so we have 46 chromosomes in one cell. Just to add on one more point to the DNA, to let you know about why we call it a very complex and gigantic molecule. It is just an assumption that if we take out all the DNA molecules from, let's say, if an average human body has got 75 trillion cells, so we have 75 trillion cells and one cell has got 46 chromosome, that means we have 46 DNA. And then if you stretch all those DNA molecules of the human body, the overall length of that will be twice the distance of Earth to the Sun. So that's long is the overall length of the DNA molecule if you take out from all the cells of the human body, and that indicates how sophisticated is that supercoiling mechanism by which the DNA molecules are confined within the nucleus. So chromosomes I was talking about, they are specifically for species number, and every species has got a specific number. And chromosomes are the storage place of all genetic information. In a normal human cell, the DNA contained in the nucleus arranged in 23 pairs of the chromosome, like 46, and then there are 23 pairs, out of which 22 pairs are autosomes, and the last pair is being called as the sex chromosome, which determines the sex of an individual. So if it's an XX, it's a female, and if it's an XY, it's a male, right? Now, gene. Now, the gene can be defined as the basic unit of inheritance. It is a specific set of genetic codes. It is a segment within a very long molecule of the DNA with specific instructions. Like if we talk about the DNA, there could be thousands and thousands of the genes on the DNA. And that one gene will particularly refer to one specific product, one specific protein. So the basic unit of inheritance is called the gene. The genes are located on the DNA. As I said earlier on, I showed you the picture on the very first slide, that the location where the genetic codes are there would be called as the gene, and there are some spaces between the two genes called intergenic region. So that was actually the DNA molecule. So the DNA molecule will have, it's a long strand. Specific instructions are there in the form of different genes on that, and that genes refers to different proteins. So genes are located on the chromosomes on its place called locus. So actually DNA is lying on the chromosome. Genes are actually on the DNA and the DNA is lying on the chromosome. Okay, so what is the general structure of the nucleic acid? The DNA along with the RNA, because when we call the nucleic acid, we are talking about both DNA and RNA. The DNA and RNA are long chain polymers of a small basic repeating units called nucleotides. And we have studied about nucleotides in our first lecture. Each nucleotide is composed of a base, sugar, and a phosphate group. The sugar could be ribose for RNA and deoxyribose for DNA. And the chain extends through 5' to 3' hydroxyl group, and the bond that connects the nucleotide is being referred to as phosphodiester bond. The first time the structure of the DNA, which is a double helical structure we all know, is being found by the scientists Watson and Crick, and for that they have been awarded the Nobel Prize. They identified that there are two chains of polynucleotides held together in the form of a letter, as you can see the picture, and these two chains are held together by hydrogen bonds. This structure explains how genes engage in replication, carrying information, and undergoing mutation. We all know that there are four different types of the nucleotides based on the nitrogenous base, guanine, cytosine, and then thymine and adenine. So it is calculated that the G plus C content of a natural DNA ranges from 22 to 73%. What is this G plus C content? It's a proportion of the guanine plus cytosine present in a natural DNA. And accordingly, it's between 22 to 73%. So it varies from DNA to DNA. And that this G plus C content also affects the physical properties of the DNA, for example, melting point. Now, the reason for this is, if you all know that the guanine and cytosine, they undergo hydrogen bonding and they will have three hydrogen bonds, while adenine and thymine will undergo hydrogen bonding with two hydrogen bonds. So more the G and C content, more bonds, and that's why higher the melting temperature. As I already said, that the four nucleotides based on the four nitrogenous bases, we know that they are being classified as purines and pyrimidines. We already have gone through their names, and it's important thing to note down that the number of purines always equal to the number of thymine always equivalent to adenine, and guanine always equivalent to uh, 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 guanine and cytosine are same, and thymine are same, because of the fact that we know that there is a specific bonding, as I mentioned earlier on, that it's G and C, guanine and cytosine, and adenine and thymine. They have a double bond, and in case of cytosine and guanine, they have a triple hydrogen bond. So it's always complementary that guanine undergo bonding with cytosine, and adenine always undergo bonding with thymine. So that's why their number will be, proportionate or same to each other. Now, in contrast to the DNA, RNA is a single-stranded polynucleotide chain, and they have a couple of differences, like they do not have thymine. In place of thymine, they have another nitrogenous base, uracil, and they have a 5-carbon sugar ribose in place of a deoxyribose sugar in case of the DNA. This is the diagram showing you the hydrogen bonding, and you can see that this is one polynucleotide chain. This is another polynucleotide chain. It starts from 5' to 3'. So the next chain is 5' to 3', opposite in direction. And here you can see that we have two hydrogen bonds here, and we have three hydrogen bonds here. So it's always complementary to have cytosine and guanine and adenine and thymine. As I was saying that, the way the DNA is organized within the body is very, very important. And this process is called as supercoiling. So you can see that the DNA molecule, they are complex with proteins called histones. Histones are just like, a rod where the DNA is wounded around, around the histone proteins to make it compact. And as a result of this, the structure form is called chromatin. As I already shared with you guys, a human cell contains about 2 meters of the DNA. One human cell, that means 46 DNA, if they are stretched together, the length is 2 meters. And the DNA in the body, if we calculate this one, could stretch to the sun and back almost 100 times. So that shows that how tightly it is packed together so that it you know reside within the nucleus. This is the diagram showing you. You can see these blocks are the histone, and this black color is the DNA. You can see the DNA is bonded around. So that's how it has a small surface area where it wounds on the histone proteins. And this particular structure is called nucleosome. These histone proteins are called as DNA core. So there are different types of the histone proteins in our body, like histone H2A, H2B, H3, and H4, and they do have different sort of functions. There are different forms of the DNA as well, based on different configurations and their specification in terms of their turn, nucleotides per turn, and all these kind of information. So there are two forms which I discuss here, B and Z form. These are the two forms of the DNA that we normally found in the organism. The B form is the normal form which is predominantly present in the cell. Okay, so that's a brief intro about uh, what is gene, what is a DNA molecule, what is an RNA molecule, how the genes are located on the DNA, how the DNA is compactly arranged with the histones and then on the chromosomes, and all that sort of information. Now we jump on to our next part of the presentation. Again, remember that's an introduction. We are not going to start DNA replication in detail here because we will have that discussion later on in detail. But that's a very quick summary. So DNA replication, as I mentioned earlier, is the process by which the DNA duplicates. And the question is why it duplicates? The answer is simple. Because cell will undergo mitosis. So when you got two cells made from one cell, you need to divide the genetic material before you divide the cell. And that's why the DNA replication takes place prior to mitosis. So once your genetic material is duplicated, then the cell will undergo mitosis and you got two cells out of one cell. So that is what the process of DNA replication is. And the picture explains you that how the two strands of the DNA, which are bonded through the hydrogen bond, they start unwinding, and as a result of that unwinding, the new strands of the DNA are being synthesized on them. So one of the strands will be acting as a template, and a new strand is being synthesized on that template, keeping in mind the same logic as I said: guanine base pair with cytosine, and adenine base pair with thymine. Here is another very important thing to discuss, that we know that DNA resides in nucleus, but there is some portion of the DNA that do exist in mitochondria, and that is being called as mitochondrial DNA. Now that's quite surprising, that why there is a need of a DNA in mitochondria, because the DNA in the nucleus, we know that fair enough that it has got genes on that, and the genes encode proteins, and that could be hormones, that could be enzymes, that could be any other thing in the cell which is needed. So that's why if we need any kind of protein in the cell, it has to be synthesized within the cell, and for this you have genes on the DNA, and the DNA is in the nucleus. But this mitochondrial DNA is doing the same task, but it's specific to mitochondria. The reason for being is that the mitochondrial membrane is uh, not permeable to most of the molecules. So there are certain chemical processes, there are certain chemical reactions going on within the mitochondria which requires enzymes. And these enzymes cannot come from outside the mitochondria to inside. So they have a mechanism to synthesize few of the proteins which are exclusively needed for the mitochondria. So this is what the role of mitochondrial DNA is. Mitochondria, we all know, is the powerhouse of the cell. It involves in a range of processes like signaling, cellular differentiation, cell death, cell cycle, cell growth, etc. They also have been implemented in the several human diseases like mental disorder, cardiac dysfunction, aging, etc. So you need a couple of proteins within the mitochondria for which there is specific mitochondrial DNA. Now, mitochondrial DNA, in contrast to the DNA within the nucleus, is pretty simple. You can see that mitochondrial DNA contains only 37 genes, all of which are essential for normal mitochondrial function. Out of this 37, 13 of these genes provide enzymes, which are involved in the chemical reactions within the mitochondria. Most importantly, oxidative phosphorylation. Oxidative phosphorylation is the formation of ATP. The oxidative phosphorylation will use oxygen and sugar to create ATP, which is the main energy source. The remaining genes out of 37 are there to code different proteins for making molecules like RNA, tRNA, ribosomal RNA, which are the essential part of that machinery to synthesize protein. So mitochondrial genes are among the estimated 20,000 to 25,000 total genes in the human genome. Now, functions of the DNA. We discussed that DNA is a gigantic molecule, the first choice to store and encode the genetic information. DNA is the chemical basis of hereditary and genetic variation, and DNA is the molecule that directs the synthesis of RNA, which is transcription, and then from RNA to protein synthesis, which is translation. Now, let's talk about what is genetic code. A genetic code is a specific set of purine and pyrimidine bases located on the DNA molecule, and they are symbolized by letters or alphabets to easily identify them. All the information, all the genes contained on the DNA are actually represented by four letters, which are A, T, C, G: adenine, thymine, cytosine, and guanine. So the combination of the three nucleotides, any three nucleotides, will result in the formation of a genetic code. So let's say if you have a long chain of the DNA where you got A, T, C, G, A, T, C, G, and repeated names. So any three particular nucleotides will be referred to as a genetic code. So in this case of ATCG here, the first three nucleotide ATC will comprise one genetic code, and that is being also referred to as codon. So a group of codons constitute the genetic code that can be translated into amino acids. So one genetic code or codon, which is comprised of three nucleotides, refer to one amino acid. So that means the second code refer to second amino acid, and so on and so forth. So when you got multiple amino acids formed, we know that amino acids join together to form proteins, and that's how the protein synthesis takes place. So you have a genetic codon on the DNA. From that genetic codon, there is another codon made on the RNA, which is mRNA. From mRNA, it goes to the tRNA, that is the translator RNA, and then the translator RNA will bring about that particular amino acid. So every code, every codon is specific for amino acid, and that's how the protein synthesis takes place. So here's the picture you can see. This one is the sequence of nucleotides you can see here. So we start from there, and then counting from there, the first three, then second three, then the later on and later on. So this is codon 1, 2, 3, 4, 5, 6, 7, 8, 9. And then every codon, as I said, refer to a specific amino acid. So it's amino acid number 1, 2, 3, 4, 5, 6, 7. And you've got multiple amino acids, which are the backbone of the proteins. So the sequence of the codons on the DNA are transcribed on mRNA, and then this mRNA will define the primary structure of the protein. Now, there are 64 possibilities of the codons. and we have 20 amino acids when we talk about the eukaryotic cells. So that means you have more than one possibility of a codon for an amino acid, because we have 20 amino acids and we have got 64 possible codons. Out of 64, three of them are being specifically called as, they have a specific function, but 61 out of 64 are specifically for amino acids. While the remaining three, as I mentioned here, UAG, UAA, and UGA, these three specific codons are called as stop codons or stop signals. What they do, we'll come to know later, but their name itself replicates that they have some role in stopping the process of transcription. Now come to the RNA. RNA is a single-stranded molecule, as I said. They have ribose in place of deoxyribose. They have uracil in place of thymine. Now the RNA are of three types, three classes. Messenger RNA, which is mRNA, ribosomal RNA, which is rRNA, and the transfer or translator RNA, which is called the tRNA. There might be some minor classes of the RNA also, but we are not very much interested in those type at this point. So let's start with the little bit discussion about the RNA. So in case of the RNA, as I said, it's a single-stranded molecule, and the concentration of the purine and pyrimidines, that is, the nitrogenous bases, do not necessarily equal one another. Now, this is unlike the DNA. Remember, in DNA, the content of the G and C and the content of the A and T, they were equal. Why? Because there were hydrogen bonding between the two chains. But in this case, it's not mandatory because it's a single-stranded molecule. However, the single strand of the RNA might be capable of folding, twisting, and making few hydrogen bonds to give rise to a little bit of hairpin-like structure, as you can see. And here you can see that some of the nitrogenous bases, which were complementary, they undergo hydrogen bonding, but there are certain nitrogenous bases, they're not complementary to each other. That's why there is no hydrogen bonding. So there's a loop here and there is a tail here. So this is specifically a hairpin shape of an RNA molecule, which results because some of the nitrogenous bases, they have undergone hydrogen bonding because they were complementary, while some of them were not. Now, messenger RNA, the name itself replicates that it carries the message. Now, this is the type of the RNA which is the first step to be made from a DNA in the process of protein synthesis. So the genetic codes or the genes, in short, they are on the DNA. Now these genes are transcribed on a molecule which is called mRNA. So mRNA is a copy of a particular portion of the DNA which we need to translate. And that's again complementary according to the sequence of the nitrogenous bases. This mRNA is being referred to as an intermediate carrier because it just carries the genetic information from the DNA, deliver it to the protein synthesizing machinery, and once the proteins are being synthesized, these mRNA are no more needed, so they will be hydrolyzed. So it's just an intermediate carrier that carries information from the DNA and translate that into a particular protein, and once the function is done, RNA is hydrolyzed. So this is what the mRNA is. They have a 5' region and a 3' region. The 5' is the untranslated region and the 3' is untranslated region, which is cap. So they have a typical structure that they have a cap on the 5' end and they have a small tail of adenine at the 3' end. Then we have the transfer RNA or the translator RNA. The transfer RNA, they have a specific shape as well. As you can see, it's a cover leaf-like structure. It's a specific leaf present in UK, and the shape of this, looks like that. That's why it's called cover leaf structure. Now you can see that it is also a single-stranded molecule, but you see that there are a couple of hydrogen bondings at different locations, but there are certain locations where there are no hydrogen bondings because they're not complementary nitrogenous bases. So it gives rise to a very specific shape where we have these 4 terminals, in front of us, and these terminals would have a specific function. just like any other molecule, they also have 5 prime and 3 prime end. You can see this is the 5 prime end. So it goes like this, and then comes to 3 prime. So it's basically a single stranded molecule, but it has undergone a specific arrangement based on the complementary hydrogen bonding between the nitrogenous bases. So there are some important structures. This is called the D loop. This is called the T loop. This is the amino acid attachment terminal, and this is the anticodon loop. All of these loops would have specific functions, would have a specific role. But the most important thing is that this particular portion of anticodon is the one that read the message on the mRNA, because mRNA carries the codon, and as I mentioned, that codon refers to a specific amino acid. So the tRNA has this property that it reads the codon on the mRNA and then bring about that respective uh, amino acid. The amino acid is being attached at this 3' hydroxyl end. And that's how you can say that transfer RNA or translator RNA is like a reader. It's like a reader molecule which reads the genetic information on the mRNA and then accordingly bring about the specific amino acid which is needed to synthesize the protein. Now, the third one is the ribosomal RNA or the rRNA. Now, these are more of a structural RNA. The ribosomes, which are located in the cytoplasm, they are free as well as they are attached on the Golgi apparatus. They are actually the protein synthesizing machinery. So the whole process of the protein synthesis occurs at the ribosome. So ribosomes, they are actually composed of rRNA and a little bit of protein. So proteins and rRNA combination give rise to ribosomes. And that ribosome, as I said, is a site where that whole process of protein synthesis takes place. This is a picture of a ribosome, and you can see that this is mRNA with a pink one. This is transfer RNA, which is having anticodon bound with the codon, and this whole molecule is the ribosome that contains, the rRNA. So that's how this mRNA is going across like this. The transfer RNA is reading this and bringing the specific amino acid like methionine or any other, and that's how the different amino acids get attached and results in the formation of the protein chain. So as I said again, we are not going in the details at this stage because we have to go through the transcription and translation in detail later on, but that's just an overview of how it goes. The ribosomes, as I said, composed of rRNA along with protein in it. So there are different categories of the ribosomal RNA. They consist of two subunits, a larger unit of 60S, which holds RNA 5S, 5.8, and 28S. This S is actually sedimentation coefficient. It's a constant. Depending upon the size of the molecule, they have different sedimentation coefficient. So 5S, 5.8S, and 28S are their sedimentation coefficient, and when they all together combine, that's how they make a 60S, and 40 part is the protein. And a 40S subunit contains 18 and 30% of the protein, which come together to form overall as 80S. So in case of the eukaryotes, there are two portions, 60 and 40S separately, and when they are merged together, overall they become 80S. While in case of the prokaryotes, it's 70S ribosome. So it's the difference between the eukaryotic cell and the prokaryotic cell in terms of the ribosome and their sedimentation coefficient. The next term to go through is about polysomes. What are polysomes? This is a specific characteristic of the eukaryotic cell. And because in eukaryotic cell, the mRNA is very complicated molecule, it's a big molecule, and it doesn't fit on one ribosome. So that's why there are many ribosomes attached together in order to provide a bigger platform where the mRNA molecule can be properly translated. So this combination of the ribosomes will be called as polysome. Now you can see here, if this is the entire mRNA molecule, you can see it's a long molecule, and it requires many ribosomes to align together to provide the platform for this mRNA molecule to be read. So that's why we call it a polysome, and this is an electron microscope real picture showing you how the polysomes look like. Finally, we come to the protein part, which is translation. Proteins, we all know, are the building materials of the cell, the final product of the gene's translation. They are the polymers of amino acids linked together, and that sequence is being actually present on the DNA, which is being, as I said, transcribed on the RNA, and then from RNA to the proteins. Proteins have a wide variety of functions, and I'm not going into the detail of the functions because you already know about the functions of the protein in the body. So there are four levels of the structure of the protein. It starts from primary, secondary, tertiary, and quaternary. Again, I'm not going into the detail of this because that's part of first year course we studied about these level of the protein structure, and you know that what is the difference between primary, secondary, tertiary, and quaternary structure. So this is a diagrammatic illustration of these four levels of protein structures. Gene expression, what we were studying till now was actually gene expression. What gene expression means how the gene can be expressed. In other words, the expression of genes is actually protein synthesis. So gene carries a set of information, and when it is expressed, it gives a product, and that product is a protein. So gene expression can be divided into two stages as transcription, which is the synthesis of RNA, specifically mRNA, and then translation, where mRNA is translated to give the final protein. There are different control levels, like because it's a complicated process of the entire process of transcription and translation. It's a complicated process, and come to know later on when we go in the detail. So it requires to be regulated. Like every metabolic cycle is regulated, so it's kind of a process which requires regulation, and there are different levels at which the regulations can be done. So some regulations are done at the level of transcription, which is formation of mRNA. Some regulations are being done after the transcription, called post-transcriptional stage, where mRNA is already synthesized. Then comes the translation. So some regulations are at that translation stage where the protein was synthesized. And then finally, some regulations are being done at the post-translational stage where protein has been produced, and then protein will undergo certain changes. Very quickly going through last few slides, just giving you an idea because we were studying about molecular biology, so you must be aware of the fact mutation. Mutation is any kind of alteration, any kind of change in the genetic information. It could be minor to the gross. It could be in the gene. It could be on the DNA. It could be on the chromosome. That can be called as mutation. So gross mutations are normally the bigger one that happens at the chromosomal level. Like you might have heard that in different clinical conditions, one of the chromosome is missing from some people. So that's a gross mutation where the entire chromosome is missing, or a part of the chromosome is missing. And there are certain mutations where certain genes are missing, and there are certain mutations where a particular portion of the gene is missing. So it starts from the mutation of a single base pair to the gross mutation of the chromosome. That entire thing is being referred to as mutation. So mutation could be autonomous or it could be induced, depending upon, like it's a natural process as well. During the replication process, the mutation can occur, and because of certain chemicals, certain radiations, or certain processes, we can induce mutation as well. So that's why we say that mutations can have harmful or can have the beneficial or can have a neutral impact. Like sometimes little mutations takes place and it doesn't affect on the final product, that's protein. Sometimes the mutations are harmful because that results in genetic disorders like sickle cell anemia or any other. And sometimes they are beneficial because we did that in order to avoid certain clinical condition. So it has all these ingredients. The pictures indicate here that if there is only one base pair mutation, we call it point mutation. If this particular CD is removed, that's called the deletion mutation. If the location is being changed, called translocation mutation. And then there's a reverse of the sequence. You can see CDE become EDC, that's called inversion mutation, and so on and so forth. There are many types of mutations. Now these mutations could be on the autosomes, these mutations could be on the sex chromosome, and depending upon that, if there's a genetic disorder, it could be an autosomal genetic disorder or it could be a sex linked genetic disorder. These are common tools in the molecular biology. I'll not go in much detail, but just to let you know and to make you familiar of the term, like polymerase chain reaction. You all guys have heard of this PCR nowadays with the COVID-19. Every COVID-19 testing, methodology mainly involve this PCR technique, PCR polymerase chain reaction. Then we have hybridization, molecular cloning, nucleic acid enzymes, DNA sequencing, electrophoresis, southern blotting, northern blotting. These are all the different techniques and tools that are used in molecular biology. Uh, Human Genome Project (HGP). Just to let you know, it's not that important. It's a big project which was actually made to collect all the genomic information on the human DNA. And it was a big project because human genome is very complicated. So that project has extended up to years and years, and now we have the genome database, which I mentioned in the earlier slides, where we have all the information of human genome, which particular site has got the genes and what kind of genes they are, and that will definitely help us a lot in order to identify the problems behind the genetic disorders and diseases. Now, finally, uses of molecular biology. It's a very advanced, discipline, and it has various uses. Like as I said, PCR is one of the diagnostic method used for, diagnosis of many diseases. Then there are, the identification of the malignancies, the cancers, the presence of genetic diseases, transplantation, forensic sciences, and there are many other uses like written here, like genetic engineering that has been used widely in the plants, and now it's being used in the animals as well. Then we have, DNA fingerprinting and pre and postnatal diagnosis of diseases in the kids, gene therapy, drug design, and all this kind of big terms. They are all the benefits of molecular biology. So we'll be ending here, and with this, the introduction to the molecular genetics is complete. If you have any question, please ask the question, and just raise your hand, and I will ask you to let me know the question, maybe on the chat box or whatever you want. So you can ask the question now. I think you guys are sleeping. That's why you are not asking the question, but that's fine. So I'm going to stop the lecture here, and we'll be having our lab on Monday at 12. So be there on time. Time is key for me, and that's why I haven't allowed some of the people enter the class today because they came late. So that's why it's not a good practice. Be on time. Nimra, you have a question. You can ask the question in the chat box if you want.
अस्सलाम वालेकुम सर। सर 12 बजे
Walaikum.
हमारी फिजियो की क्लास होती है, लैब होती है, तो हम अवेलेबल नहीं होंगे।
Uh, just hold on. Let me